Hello, my name is Sofia Tseranidou and I'm going to present you my work regarding the Nucleus Pulposus cell network modeling of normal intervertebral disc. Low back pain is the leading cause of disability worldwide, affecting approximately 80% of adults at some point during their lifetime. One of the main causes of low back pain is the intervertebral disc degeneration. Intervertebral disc is the largest avascular organ in the human body, located between the vertebrae of the disc and consisting of three mainly hydrated tissues: the nucleus pulposus, the annulus pulposus, and the cartilage and plate. Its degeneration might be induced due to genetic inheritance, the age, the sex, biochemical factors, and imbalance in the metabolism. Due to the avascular nature of the disc, the nutrient supply of the nucleus pulposus is made by diffusion through its dense extracellular matrix. Looking into a non-degenerate IVD, we will see that the anabolic and the catabolic processes that regulate the extracellular matrix of the disc are kept in balance. If this balance is disrupted in favor of the catabolic processes, then we have the degradation of the matrix, causing the IVD degeneration, which might lead to low back pain. Until now, the treatment of the degeneration is mainly based on surgery procedures and conservative approaches. Accordingly, a knowledge-based regulatory network model of nucleus pulposus cell for normal IVD regulation is presented. The network was built from a unique curated corpus of 130 journal articles in IVD research. Proteins are represented by nodes that interact among each other through activation and inhibition entities. Through a fast interpolation of Boolean rules, the semi-quantitative steady stage of the regulatory network model, or node activations, were calculated. Simulation tests evidence the limited literature knowledge to represent normal IVD and guided corpus enrichment through the string database. Afterwards, we have assessed our networks against independent experiments. And finally, we have performed a full factorial sensitivity analysis in order to identify which of the 15 cytokines and 4 growth factors was affecting most the structural proteins and degrading enzymes of our network. The enriched regulatory network model represented successfully an anabolic basal steady states, as we could expect in a normal IVD, and was able to replicate 2 independent in vitro experiments. Interleukin 1 beta and TNF-alpha were promoted catabolism, while TGF-β was not able to regenerate the disc. From the sensitivity analysis, we identified TGF-β and interleukin receptor alpha as the most powerful rescue mediators proteins that have been proposed before for treatment of intervertebral disc degeneration. To sum up, the present regulatory network model was successfully converted into independent in vitro experiments and stands for a unique model to integrate cellular protein signaling at the tissue level and explore the degeneration onsets. Thank you very much for your time.